You guys done? All right, why don't we just go over it again? How'd you do?
About half.
Okay, good. Okay, question 1. Which intermolecular processes primarily derives from the formation of bilayer when phosphates are into the water? Did you see the correct answers there, or just so you know the answer already? So the idea here is that like nonpolar things are not really attracted to each other. They're not really binding to each other. Water's excluding them. So the biggest energy problem is when water tries to dissolve these things, because it breaks up the hydrogen bonds between water. So the reason that they formed this arrangement is because then water gets to be in its lowest energy conformation. So that's the way you want to look at that one.
Can you, sorry,
Mhm.
I kind of understand that. Wouldn't the first one kind of be true too? Because it's more, I guess you're talking in more of terms of it's more favorable for the water
Yeah.
than the lipids, or?
You know, honestly, this doesn't sound that bad either. The ordered arrangement of the bilayer is more favorable than the disordered state of individual free-floating liquids. I think this is also true. This is more true, I guess, because it focuses on the water problem. Yeah. A lot of questions are like that, you know?
Yeah.
Which statement best describes how cholesterol affects membrane fluidity? So I always say it acts like antifreeze, right? So then it's going to decrease fluid at a high temperature, increase at low temperature. Compared to a typical animal cell membranes of the polar bear. So what are we looking at here? Why are we saying this?
I didn't know if it was talking either about like contact with like a water while they're swimming or cold when they're walking.
Yeah, I think it's the cold.
Yeah.
Unsaturated.
Yeah.
Unsaturated, because then they're going to be a little more, the membranes be more fluid. Yeah, they're not going to be taking in water through their feet. Potassium channels, that's just kind of crazy. So it would be one of these two, and you want to go with the one that's going to be more fluid at cold temperature.
And why is it that when you have double bonds in the lipids that it's more fluid at cold temperatures?
Right, because the phospholipids don't stack as well.
Yeah.
So they're going to be more likely to be fluid.
Okay.
Yeah, and that's why, like homeotherms, we've got a warm internal temperature, so it's more saturated. You know, like plants are polyunsaturated because they are, at colder temperatures than us. Small question.
So they have unsaturated phospholipid, and then because of the cold, it works kind of like saturated
Yeah,
phospholipid.
yeah.
All right, yeah, because I thought it was going to be saturated phospholipid too, so that it would have more protection. But
Pretty sure it's unsaturated, but we'll see in the end.
It is.
It is, okay. Which molecule diffuses through the membrane most quickly? That would be this one, because it's both nonpolar and small. Benzene's nonpolar, but it's bigger than ethylene. So there's always those two things involved, size and the polarity. Which fatty acid tail would contribute most to the stability of a cell membrane of a thermophilic. So thermophilic is going to be in what kind of
Hot.
in a hot situation, which is going to be most stable and hot. this long fatty acid tail. And again, here's your unsaturations that are going to make it more fluid. So glucose typically enters the cell through which mechanism? That's pretty easy. It can enter through simple diffusion. There are mechanisms for this, and if there's glucose in the solution, phenocylation will pick it up, but this is usually it's just going to be facilitator diffusion. Which statement represents a notable difference between simple and facilitated? That would be this guy, right? More so because the other ones are wrong. Facilitated doesn't use ATP, can happen in any cell, unlike simple facility can transport ligands against, nope. Facilitated is still passive, so it has to be with the concentration gradient. Rate of osmosis across the cell can depend on intracellular, extracellular, polarity, molecular weight, presence of aquaporins. Definitely 1 and 2. Which ones have 1 and 2? Only these two. So either all of them or this one. Pretty sure the molecular weight doesn't matter. The polarity definitely doesn't matter. It's just the number of solute particles, not the polarity. In nerve cells, sodium potassium ATPases, what's the primary purpose? You're restoring energy. Right? Here's a thing too, to increase the concentration inside the cell, no, because you're pumping it out. Increase potassium outside the cell, no. Your physiology is going to control the outside of the cell. Cell can only control what's inside. And action potential, sure, but not in every cell. Right? Only some cells do action potentials. Newly developed medication for pulmonary hypertension targets blood vessels in the lungs, but does not affect blood vessels in the liver. Which of the following is most likely true? Ligand is specific for lung and liver vessels, but which is metabolized? No. Ligand is specific for blood vessel receptors in the lung, but not in the liver. Possibly. Receptor is upregulated? No. Receptor is only expressed in the blood vessels in the lungs. I'm going to go with this one. Ligand is specific for, why not this one? It is a receptor. Oh, no. Medication is not a receptor. Okay, so it has to be that one.
So yeah, so that's like kind of the thought process I went through. I ended up clicking the ligand one, but couldn't the receptor kind of also be true if you weren't, like I know they're specifically talking about the medication, but
Yeah.
couldn't the receptors also play a role in it?
Definitely. Yeah, if the question was slightly different, you could have different receptors here and there. Definitely. Yeah. Which of the following is not true regarding ligand-gated ion channels? That's definitely true. Probably true. Probably true. Open or close in response to deformations? No. Those are stretch-activated channels. Once phosphorylated intracellular segment of tyrosine kinase. By the way, I get a message back from the MCAT about questions like this, and if you get them wrong, I change your grade in this. Okay, you have to get this one right. That's our mechanism there. Cytoskeleton of the cell can generate movements, cytokinesis, or propulsive forces. Which structures are likely linked to these? Okay, so cytokinesis versus flagella. So cytokinesis is microfilament and microtubules for the flagella. Intermediate filaments don't do any movement. So all three of those are out for that reason. Which cytoskeletal structure is linked with the movements of the macrophage? So macrophage is doing amoeboid movement. So that's got to be microfilaments. Might be cell crawling, but I think it's amoeboid. But either way, it would be microfilaments. Okay. Which of the following is not a reason cancer is hard to treat? That's definitely true. This is definitely true. This is true. Increased contact inhibition. So what we talked about last week is it's got decreased contact inhibition, right? And that allows them to form tumors and keep getting bigger. Which statement best describes chromosomal movement during mitosis? To bind tubulin polymers on centromeres to initiate homologous separation. No, centrioles bind to kinetochores. That's true. Centrioles send up microtubules which bind to kinetochores. That's more true. Each centrosome binds an individual chromatid. That's not necessarily true. Centrioles on chromatids. Oh, they're not on chromatids. Centrioles, okay. Retinoblastoma, RB, is an important cell cycle checkpoint protein that prevents transition from G1 to S. Presence of the cyclin CDK complex releases it from that means it binds it. Which of the following mutation scenarios is least likely to form neoplasm? Loss of RB, no RB mutation. Cyclin that cannot, well, this one then, right? Because RB is normal and the CDK is not going to inactivate RB. So this is definitely not going to become carcinogenic. This could be carcinogenic, right? And loss of function, no, definitely not. Caspase enzyme mediate apoptosis by attacking which of the following? Okay, so none of the cysteines are true. Caspase, there's a cysteine in the active site, and it cuts proteins with an ASP in them. So I don't know if I said that in class, but
Is it not involved in BCL2?
well, the thing is, once it starts, like these are all definitely wrong. So you stop looking at them after that. You know what I mean? You want to keep moving. Which type of membrane receptor protein best describes receptor tyrosine kinase? Or enzyme receptor. Which signaling molecule least likely serves as a ligand for an intracellular receptor? So it's going to be intracellular, has to be something that gets into the cell, nonpolar, 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 polar. Which, when acetylcholine binds to disruptor, oh, this is a little tricky too. This is an example of what? A ligand-gated channel. Now, what's tricky about this is it is the nicotinic receptor, but the question is, it's an example of what? So you have to go with the more generic answer.
Actually, I was so
Yeah.
confused.
Yeah,
I ended up with the right answer, but I was like going back and forth. I was
Yeah.
thinking that.
That's
Right.
what I was confused on.
So nicotinic receptor is a ligand-gated ion channel.
Yep. And it's activated by acetylcholine. Steroid hormones such as testosterone and estrogen act on target cells by passing. If steroid hormones can enter any cell, how do they only produce effects on target cells? This is pretty easy. Only target cells have the intracellular receptor. All cells have the same genes. Proper phosphorylation cascade, no. Enzymes the enzymes to modify, no. That one's pretty easy. When normal human cells grow in culture, they will divide a limited number of times, typically 50 rounds of mitosis. After this number is reached, some cells become apoptotic. This is dealt as a result of C, or telomere shortening. That's the Hayflick limit. Replicate copies of each chromosome are called sister chromatids and are joined at the centromere. It's a little confusing because really the kinetochore is the proteins around the centromere. Sister chromatids are attached at the centromere. Which of the following is not a method of post-transcriptional control of eukaryotic cells? We didn't talk about this. See, processing the transcript, post-transcriptional, so that's true. Digestion of the DNA immediately, what? What? Selective degrading of mRNA transcripts, that happens. Selecting mRNA molecules that are translated. That must be true because this is definitely not. Proteins that stay within the cell are produced, and again, we didn't cover this in class, but they would be in free ribosomes. Because the ribosomes attached to rough ER are only for secreted and membrane proteins. That way the ribosomes are right in a membrane and you get post-translational insertion. We didn't go over that, but.
What about the nucleolus?
About the what? Nucleolus?
Yeah.
No, nucleolus
And
only makes ribosomes.
Yeah.
That's its only role. Which increases the fluidity of the plasma membrane? Double bonds? Yes. Tight alignment? No. Cholesterol increased fluidity depends on the temperature. Having a large number of membrane proteins? No, I think that would make it more rigid. So we're going to go with double bonds because I know that's true. Sucrose cannot pass through the membrane of a red blood cell, but water and glucose can. Which solution would cause the red blood cell to shrink the most? Shrink would have to be hyperosmotic. And since glucose can go through and sucrose can't, this is the one that would be cause the most shrinkage because glucose going through would change that the osmotic balance between the inside and outside. So it would not shrink as much. Which of the following statements best describes the sliding filament mechanism of muscle contraction? Actin and myosin filaments do not shorten, but rather slide past each other. Sounds right. Actin and myosin shorten? Nope. Actin filaments shorten? Nope. Neither filament shorten. So that's pretty easy. What's the role of calcium in muscle contraction? Binds to the troponin complex and control myosin to move over, revealing binding sites for cross bridges. That sounds right. Binds tropomyosin, binds to troponin, enabling tropomyosin release ATP. Nope. Let's see how I did. See, this is the key to the MedCat. You write all the questions yourself, and then you get them all right. I got all these questions from not Kaplan, Khan, and the Khan Academy.
On the last one we just did, it says that mine was wrong, so I just want to make sure that because we selected the same answer.
This one
Yeah.
binds to troponin, enabling
Oh, wait.
tropomyosin.
No, sorry, I didn't select that. I swapped the tropo.
Okay.
Can you remind me what tropomyosin does?
So tropomyosin is the long coiled coil that hides all the myosin binding sites. So when troponin binds calcium, tropomyosin moves and then myosin can attach.
That's like
Thank
that
you.
caulking thing, right?
Yeah. Grabs and moves. Let's go. All right. Is that good? That's about all I have for today, unless you want to talk about the exam. Did you guys take the exam yet? Cowards. Cowards. So if you want to talk about that material, we can, but if not, this is your time. Jay and David, any questions? Did that go okay for you guys?
Yeah, it went pretty well.
Yeah, no questions so far.
Okay, good. All right, well, that's all I have for today. If you don't have any questions, we're gonna call it a day. Okay, see you on Wednesday. We'll do a similar thing with physiology. Maybe I'll come up with more questions. We'll do a little longer